the next challenge on our agenda. We have monoclonal antibody-based therapeutics for new variants of emerging viruses. And we'll have two presenters on this topic. We have Christian Wimmer from Hera and also Mani from DG RNI, who is going to discuss the details of the call. I guess Mani is going to start.
Sorry, I'm going to try and load up the presentation first, and hopefully you can see it. Is that visible now?
Yes.
Perfect. So actually it is going to be Christian who's going to start and kick things off and provide you with a bit of the policy context before I move into a presentation about the call specific aspects. So Christian, can I ask you to take over and I'll help you with the slides as we go through.
Yes, whatever you mean with taking over. So if you advance the slides,
I'm trying to.
Helpful.
Seems to be a time lag, but there we are.
So good morning, everybody. My name is Christian Wimmer. I joined Hera, the Health Emergency Response Authority, 2 years ago. So at the early inception phase. Hera, as you know, I'm sure you know, was a reaction to the COVID pandemic, where it was clear that the response in member states and at EU level was strong, but that there was room for improvement. And for this purpose, Hera was set up in order to address a number of shortcomings. So I would like to put this topic on the monoclonals in a wider context of why is it important for Hera. So on the next slide, you see a summary of the main tasks of Hera, which is split into a preparedness mode, in which we are currently in, and a crisis mode. So crisis mode would be declared by member states as following an assessment of an outbreak of a new pandemic. At the moment, such a declaration has never been made. So we are in the preparedness mode, which allows us to get ready for the next pandemic, which involves a number of core tasks, which is threat assessment and intelligence gathering. So these are two levels of intelligence gathering. On the one hand, upcoming emerging pathogens on the one hand, but also a look into supply chains of medical countermeasures. Then my focus in Hera is advanced research and development capacities for medical countermeasures. And you see, I'm using now medical countermeasures a second time, and I will use it quite often. Medical countermeasures are the only tool for pandemic preparedness Hera is supposed to develop. So medical countermeasures can be therapeutics, can be vaccines, diagnostics, but also protective equipment. And Hera, of course, wants to make the products, the countermeasures, available. But of course has to take into account what exists and what does not yet exist. So it's the full value chain, which can start in case a medical countermeasure is available to stockpile it. If not available, it would be a procurement an option. It depends on the individual situation, but also looking into supply chains in case production need to be scaled up. So this is pandemic preparedness, that we think this through. And then medical countermeasures might not yet be available. So the question is, where do we start? In which technological readiness level can we take off. So it might be very low, but ideally would be advanced. Now I will not go into the details of the crisis mode, but I think that's anyhow my last slide. The next one to place this work in a context of funding. So, oh no, sorry, this allows me to introduce you what we have done in the past 2 years in Hera. So there's a threat prioritization exercise on the upper left corner. The communication introducing Hera identified already viruses, CBRN, and AMR, antimicrobial resistance, as the priority threats for Hera. This was further elaborated by identifying 10 viral families for the agents with pandemic potential, by identifying antibiotic-resistant bacteria under AMR, and a similar exercise for chemical, biological, radionuclear cross-border health threats. So you see, it is a lesson from COVID, but the scope of Hera is beyond the management of pandemics. Although it's at least the focus of my work, which included also to identify a list of critical medical countermeasures, doing a gap analysis, which brought us to the threat prioritization. There's work going on to set an IT platform for intelligence gathering. Here's the idea, the keyword is the interoperability. So a lot of information is currently being gathered, and it's critical to bring this together in order to have the full picture. And Hera set up a laboratory network, which are already quite active, for example, looking into mpox viruses, so ongoing work. Hera is also engaged in the wastewater surveillance beyond the COVID-19 surveillance. So when I talk about Hera, of course, Hera is doing this in collaboration with other Commission services, DGs. For wastewater, the Joint Research Center is of utmost importance. There are strong collaborations with at least the DG GROW, DG Santé, DG RTD. But also Hera takes a global look, because a pandemic is a global threat, and thus requires global solution. So Hera is active on many fronts. We have currently 90 colleagues working on improving pandemic preparedness. And now with the shortages communication, Hera is even taking up additional tasks in addressing shortages of medicines. So now let's move to the next slide, which clearly shows that the importance of the Horizon Europe program for the Hera work program. So Hera was set up 2 years ago. This means it was in the middle of the financial framework. So Hera has not an own budget, but existing budget lines were reallocated to allow Hera to become active. This, of course, was also very useful, because we could already take advantage of existing structures and develop an ambitious work program for Hera. So the main funding pillars are Horizon Europe, EU for Health, and RescEU. RescEU, this is the stockpiling. EU for Health covers a wide range, including advanced research and development, and Horizon Europe. So I circled a few elements of Horizon Europe that are of highest importance for Hera. So let's start with research infrastructures, very important for pandemic preparedness. We have a project called Isidore, which is already setting up infrastructures. European Research Council bottom-up approach, but a very important research project are there carried out currently with regard to pandemic preparedness. But the main funding source for Hera is the health cluster under Pillar 2, where we have a very ambitious work program for 23 and 24, and are currently working on the work program for 25. But also I mentioned the CBRN, so Civil Security Cluster 3 is important. And this brings me now to Pillar 3, to the European Innovation Council, EIC, is because of its focus in the or the possible focus on advanced development is in as much as important as it is probably the pillar that brings the first tangible results forward. So we had 2 topics under EIC. So there's a very good collaboration between Hera and EIC on challenges. So there was one on a challenge to avoid social distancing by technical methods, be it clean air, be it membrane technology, which I cannot share only good news with you, which unfortunately did not bring forward a successful project. And the topic we are discussing in this session is on monoclonal antibodies. So this complements the work program 23-24, which addresses vaccines, which addresses reaction to immunization, diagnostic devices. So we have many elements in this work program, also in EU for Health, except one, and this is on monoclonal antibodies. So this is an important start for Hera to enter this area, and I'm very grateful to the colleagues in EIC to have maintained this topic in a hard competition in the work program. So monoclonal antibodies turned out to be very successful in the COVID crisis. There's one success story where within 90 days a monoclonal antibody was identified and brought to GMP production level that could be used for the treatment of COVID. Monoclonal antibodies also open the pathway to diagnostics. Monoclonal antibodies also are a starting point for vaccine development because they allow reverse vaccination. So instead of running in silico models on potential vaccine targets, monoclonal antibodies tell us already how the human body reacts to a given virus. And brings us to another element of the puzzle for pandemic preparedness. So I noticed that the focus of this topic is on monoclonal antibody-based therapies for new variants of emerging viruses. Very important starting point. I will not go into the details. I leave this to Mani of the call text, but when we talk about new variants, of course, this implies that we are looking for broad spectrum monoclonal antibodies. This is always this is the most preferable situation. But I understand that this topic is also open to platform development, to novel approaches. So it is a very well balanced, wide spectrum of work towards monoclonal antibodies for major environmental threats, and of high importance for Hera. And having said this, I would like now to hand over to Mani so that we can go into the details of the call. Thank you.
Thank you very much, Christian. Thanks for providing that kind of wider policy context into which this call from the EIC fits. So quite simply, this call in the kind of the era of pandemic preparedness and precision medicine is looking to support kind of strategic approaches leading to broad spectrum monoclonal antibody-based therapeutics against new variants of emerging pathogens of high concern. So it's very much seen as a line of defense, which is complementary to new vaccines. And the call, as Christian just alluded to, has been developed in partnership with Hera by the EIC Program Manager for Health and Biotech, Yordanis Azimanoglu, or Danos, as he's known to us, who I'm sure many of you know. And I'm also pleased to say that Danos is with us to help answer any questions you may have on the technology and the scope of this particular call. So, as many of you know, in recent, whoops, I seem to have jumped 2 slides there. So, just to come back to the kind of the background and the context of this call, as many of you know, in recent years, monoclonal antibody products have dominated sort of the biotherapeutics markets with hundreds of novel mAbs and mAbs-like proteins now in clinical development for the treatment of diseases, including in areas such as oncology, which is another area in which Danos is, of course, focused on. Now, classically, mAbs are immunoglobulin G molecules, but advances in recombinant biotechnology, protein chemistry, combined with sort of a greater understanding of mAb structure and function, have also led to a kind of a growing interest in sort of recombinant varieties of mAbs. And the interest in a lot of this stems from the ability and potential for mAbs to offer a number of sort of advantages in terms of the production volumes, which Christian alluded to, in terms of formulation and clinical advantages, including sort of improved production yields and general stability. The potential for alternative use to administration, which, as you can imagine, in the case of an outbreak, could be quite advantageous in terms of the response rate, as well as kind of enhanced functionality and activity. So moving to the kind of the scope of this call, sorry, I'm actually I'm jumping ahead, both in slides and in terms of content. And importantly, these therapies also offer options for individuals with immunodeficiencies, older people, and or individuals with polymorbidities, as well as immunocompromised individuals with poor or no response to vaccination. Further, sort of if you look at it in contrast to engineered therapies, the process of scaling mAb production from R&D lab to the commercial production facility is also relatively well defined, both in terms of the platform technologies that are involved and the regulatory pathways. So again, these are things that Christian alluded to in the context of the COVID response. And what this can mean is that a facility can, in principle, be switched on with sort of minimal lead time, enabling rapid availability of product in the event of an outbreak. And their potential is such that the World Health Organization is, for example, working on mAbs in areas such as HIV, influenza, and there's also quite a lot of research which Christian touched on in the context of the use in infections such as malaria. So what's the scope of this call? So it covers the development of broad spectrum monoclonal antibodies. This acknowledges a kind of a key challenge we must factor in terms of our ability to manage outbreaks and respond to the constant evolution of viruses. The focus on broad-based therefore refers to both the ability to respond rapidly to the evolution of variants that could prolong the outbreak and the resultant impacts, as well as the type of outbreak. So it's not just about COVID, it's about all types of potential future pandemics and our response to them. A second dimension we refer to here is efficacy, and the efficacy of these type of mAbs-based treatments in being able to respond to variations between the response of individuals to the treatment. And then finally, given the focus of on outbreaks, a key dimension is also on the administration. So approaches such as extending the half-life of the antibody or injecting mRNA coding for mAbs could simplify production or the administration of these therapies, which will, as you can imagine, be important as part of our response to dealing with such an outbreak. As with the other calls that you've been hearing about, and we have one other to follow, we have 50 million euros in place. And what we're fundamentally trying to do here is to enhance and develop a European capability to respond to future pandemics, have the capability for rapid detection and analysis of virus variants, which again, touching on what Christian presented, using a MABS approach can also inform vaccine development, to integrate our capabilities, such as the Hera incubator, which you heard about, with relevant international systems and networks to target the variants of highest concern. And finally, to develop a platform of approaches that can ensure efficacy of future treatment. So hopefully that's given you a bit of context, both to where this challenge is coming from and what we are looking from you is as part of response to this call. So with that, thank you. And together with Danos and Christian, we'll be happy to take any questions from you on this call. Thank you.
Thank you, Mani. Are there any questions?
So as of right now, there's a procedural question, which I would, I think, turn to Giovanni. The question is, what sort of documents are needed for the applicant to show the 30% co-financing now that the equity component can't be used?
So, thank you, Jonas. No, there is no specific document the applicants need to provide. Still, it's important to say, provide, on the contrary, as much information, explanation, and documentation as possible to explain and to convince the experts that this 30% is at disposal of the company. So there is no specific, I repeat, no specific template or document, but it is very important to provide some solid, information that would help the experts assessing this element.
Thank you. And for clarification, this question came from Anna. Currently, there are no questions on.
Okay.
No, indeed, just this moment for you, Mani, I assume. So, just for everybody, I kindly remind you to go to slido, and then go for the hashtag accelerator_challenges. So you can turn in your questions, and this has just happened right now. So the question is, in which clinical status does this challenge expect as starting point?
Can I ask Danos to come in on that question, please? I think you're muted at the moment, Danos.
Okay. It seems that for some reason Danos is either not hearing us or so we will.
Can you hear me now?
Yeah.
Yes, please go ahead.
Thank
Should
you.
we
I'm sorry
repeat the
for
question?
So to get everybody on board, the question is, in which clinical status does this challenge expect as a starting point?
All the accelerator challenges are usually addressing the first clinical development, clinical trials, early clinical trials to show efficacy. Mani described very, very sufficiently the different angles that we're trying to pursue here and what we want to cover. What is acceptable at the accelerator level is, of course, to ask for funding for late validation stages and early clinical trial evidence. Both are needed for the development of monoclonal antibodies. For instance, he mentioned that we want to address the inter-individual variability, which is a very important issue. Or he also mentioned another need that you want to use it as an outpatient, being an outpatient in a clinic, taking outpatient services, or all these require clinical work. But at the same time, when he described the scope, money, he noticed that we want to address novel solutions in the platform development of monoclonal antibodies, in general, the next generation monoclonal antibodies. So that would also be consistent with some validation of preclinical work before you enter the clinical level of work. What I would like to add here, taking advantage of this question, is the following: that the broad spectrum monoclonal antibodies, which is the cornerstone, the center of this call, it's actually the broad spectrum of high concern, as Christian alluded, and money. They do belong to what is called the next generation monoclonal antibodies. What people sometimes forget here or disregard is, yes, the monoclonal antibodies have been around for decades, but the next generation is very much needed now. And part of this next generation are the broad spectrum, for which I think you heard by the two speakers why they're needed.
Okay. Thank you.
Thank you. There are no more questions, but Anne-Marie just shared the slide where you can turn them in.
Yes, you can use the QR code also in order to help you use Slido for questions.